What are enzymes? Enzymes are specialized proteins that act as biological catalysts in living organisms. They speed up chemical reactions without being consumed in the process. With an enzyme present, the same reaction can happen much faster, sometimes millions of times faster. Each enzyme has a unique three-dimensional structure with an active site. This is where the lock and key or induced fit mechanism comes into play. The active site binds specifically to certain substrate molecules. Without enzymes, biochemical reactions would require high activation energy. Enzymes lower this activation energy, making reactions happen much faster at normal body temperatures. Without enzymes, most biochemical reactions would happen too slowly to sustain life. Chemical reactions in our bodies would be thousands to millions of times slower. With enzymes, these reactions happen rapidly, allowing organisms to respond to their environment and maintain life processes. Examples of enzymes include amylase in saliva that breaks down starches, lipases that digest fats in the small intestine, and DNA polymerase that helps replicate genetic material during cell division. Now that we understand what enzymes are, let's explore how they actually work. Enzymes work by temporarily binding to substrate molecules at their active site. This binding creates what's called an enzyme-substrate complex. The binding causes changes in the enzyme's shape, called induced fit, or creates strain in the substrate's chemical bonds. The enzyme then facilitates the chemical transformation, converting substrates into products. After the reaction is complete, the product is released, and the enzyme is ready to bind another substrate molecule. Several factors affect how efficiently enzymes can catalyze reactions. Temperature greatly affects enzyme activity. Each enzyme has an optimal temperature range where it functions best. pH levels also impact enzyme function by altering the enzyme's shape and charge distribution. Most human enzymes work best near neutral pH. Enzymes can be inhibited in different ways, affecting their ability to catalyze reactions. In competitive inhibition, a molecule similar to the substrate blocks the enzyme's active site, preventing the substrate from binding. In non-competitive inhibition, the inhibitor binds to a site other than the active site, changing the enzyme's shape so it can no longer properly bind to the substrate. Enzymes are classified into 6 major groups based on the reactions they catalyze. The first 3 classes are oxidoreductases, which handle electron transfers, transferases, which move functional groups between molecules, and hydrolases, which break bonds using water. The remaining 3 classes are lyases, which remove groups without using water, isomerases, which rearrange molecules, and ligases, which join molecules using ATP energy. Enzymes are essential for numerous biological processes. They enable digestion by breaking down food, drive metabolism by converting nutrients to energy, and facilitate DNA replication during cell division. Enzymes are also critical for cellular respiration, converting glucose into ATP, and detoxification processes that remove harmful substances from the body. Beyond their biological roles, enzymes have numerous practical applications. In medicine, enzymes are used in enzyme replacement therapy and diagnostic tests. In food production, they're essential for cheese making, meat tenderizing, and brewing. Industrially, enzymes power biological detergents and textile processing. Enzyme deficiencies can lead to various metabolic disorders. Phenylketonuria, or PKU, results from an inability to break down the amino acid phenylalanine. Lactose intolerance occurs when there's a deficiency in the lactase enzyme needed to digest milk sugar. In normal metabolism, the enzyme phenylalanine hydroxylase converts phenylalanine to tyrosine. In PKU, this enzyme is deficient, causing phenylalanine to accumulate and lead to brain damage if not managed through diet.